afternoon. I'm Steve Cubison, Managing Director for the Technology Commercialization Office of George Washington University. And welcome to the first DC Startup Showcase featuring startups from George Washington and Georgetown universities. George Washington and Georgetown are the two largest research universities in Washington, D.C. George Washington will host this first event, and in future meetings, we will rotate the host responsibilities as well as potentially include other universities in the area. After some brief introductions from 2 to 4, you will be hearing startup pitches from 3 startups from each of the universities. After each 10-minute pitch, there will be a 5-minute question and answer session with our expert panel. Our expert panel is composed of representatives of our sponsors, Charlie Anders from Wilson Sonsini Goodrich and Rosati in their DC office, and Peter Weissman from Blank Rome's DC office. Wilson Sonsini is based in Silicon Valley and is the largest venture law firm in the country. Blank Rome is a prominent DC-based law firm with a diversified practice. We also have 2 outside experts on our panel, Neil Sweeney from MedTech Junction and Matthew Massaw from Epiderix Capital. Niall is an angel investor and Matthew is a venture capitalist. So we have on our expert panel legal experts with broad knowledge of company formation and patents, as well as representatives from early and late stage investing. These question and answer sessions will only take questions from the panel. So people in the audience should hold their questions until the networking session from 4 to 5, where they'll be able to join a networking room, a Zoom room, for each of the companies that will be presenting. We will also have Zoom rooms for our sponsors where you can ask them questions about startups, be that patents, company formation, or funding. During the networking session, you can hop from Zoom room to Zoom room, so feel free to wander. In the past several years, GW has held similar startup showcases, but only for GW startups. We always arrange these meetings so that we connect the 3 key elements of startup formation. That includes combining technology, people, and money. So the audience today is composed of researchers, business entrepreneurs, and funders. So please make use of this networking session and make key connections that can help you fund your company, connect with an entrepreneur that can help you move forward with your company, or find a technology or venture that you might want to get involved with. Make the most of these rich networking sessions with representatives from our 2 great universities, and most of all, have fun.
Thanks for the great introduction, Steve. Hi, this is Brian Koblitz. I manage the Life Sciences Technologies here at George Washington University. I'm excited to share this DC Startup Showcase with you today. I have a few slides to help you visualize the program that Steve described and get you ready for this pitch session. First, I'd like to encourage you to come visit us at our website, especially in these times when we can't do it in person. At the website, you can search our available technologies and learn about the processes of licensing and patenting. You can also find our contact information for our team, which is small but nimble. We try to operate at the speed of business, which is not always that common in an academic setting. Aside from Steve and myself, we have Jerry Komanescu, who manages the physical sciences technologies, and Mary Luceri, who runs the back office functions. Another new feature of our website is an inventor portal. So those of you GW inventors out there, your next invention you can submit online, and you'll be able to follow up with it as the patenting process continues and as licensing deals are made. Additionally, this year we have launched a network called GW CAN, or the GW Commercialization Advising Network. This is designed to help mentor upcoming entrepreneurs or help make connections to experts for entrepreneurs who've done it before and are just seeking a specific kind of help. We've had some great questions on there already in just a few months of activity. Hope you can come on and either get some help or contribute your knowledge and connections, or both. You can find the link to both our website and to the GWCAN site below this video on the YouTube page, right underneath the Zoom links for the networking session, which will come after the pitches. I'd like to give a quick thank you to our sponsors again. These are some law firms that we've worked with for a number of years, both in helping us educate entrepreneurs and inventors, as well as doing patent prosecution and negotiating business deals. We'll hear a quick word from Wilson Sonsini in the middle of the program. Right now, I'd like to share a few details about Blank Rome and Peter Weissman in particular, who is a GW Law alumnus and co-founder of the GW Entrepreneurs Roundtable. And Blank Rome is a full-service law firm, and Peter will be available in Blank Rome's Zoom room for networking at 4 p.m. today, after the pitches. And here are the companies that will be pitching. I'd like to highlight that we really have a broad array of technologies coming here from George Washington and Georgetown. These cover the gamut from small molecules for different therapeutic indications, as well as biologics and devices, both for surgery and as implants. Each startup will be pitching for 10 minutes, after which they will get 5 minutes of questions from our expert panel. And the experts have a broad array of experience having to do with startups and medical startups in particular, both from the funding required to get them going and keep them going, the business know-how to develop a technology and get it through the regulatory pathways, and the legal backgrounds that are needed to protect the intellectual property and do the business deals and company formation. After the startup pitch session ends at about 4 o'clock, please stay with us, but not on the YouTube channel. We'll be moving over to Zoom room so that we can see you face to face. You can ask questions directly of the companies and of the sponsors, or just meet other people who are interested in medical entrepreneurship here in DC or around the country, and as they are visiting from all over today. We'll have 9 separate Zoom rooms to choose from at that time, and the links again are below this YouTube video.
Hello, everyone. I'm Claudia Stewart, Vice President for Technology Commercialization at Georgetown. My office staff and I oversee all of the university's intellectual property, and we assist in launching startups as part of that responsibility. The research strengths of Georgetown fall into several strong categories, but the most active are cancer and neurosciences research. Georgetown's Lombardi Comprehensive Cancer Center is one of only 71 such centers designated by the National Cancer Institute. The nature of our research is reflected in our startups, most of which are focused on developing cancer therapeutics and related health innovations. One of our startups is currently developing a repurposed cancer drug for use in neurodegenerative diseases, and that technology has already completed a phase 2 clinical trial for Parkinson's disease. In fact, 7 of the technologies we have licensed to small or startup companies are engaged in phase 1 or phase 2 clinical trials. While Georgetown has had several clinical success stories, such as the whole body CAT scanner and a widely used papillomavirus diagnostic, as well as Imlygic, an oncolytic viral therapy sold by Amgen and used to treat certain unresectable melanomas, perhaps our best known recent achievement comes from one of our esteemed faculty, Dr. Richard Schlegel, who is one of the inventors of the first vaccines for human papillomavirus. That vaccine technology was used both by Merck and GlaxoSmithKline. We look forward to our future successes, some of which we hope will come from our current group of startups. Thank you for joining us, and I hope you enjoy Georgetown's presentations today.
Hi everyone, I'm Ruchika Nichara, Senior Director, Office of Technology Commercialization at Georgetown University. It is my pleasure to introduce 3 of Georgetown startup companies that are presenting today in this event. The company that would be presenting first is Tokar Pharma. Founded in 2019 by Dr. Milton Brown and Damian Dolan, Tokar Pharma has developed a proprietary tissue-targeted platform technology. One of the company's lead small molecule therapeutics, YK4250, was developed by Dr. Brown during his decade-long tenure at Georgetown University, where he was the founding director of Drug Discovery Program and endowed chair in experimental therapeutics. While at Georgetown, Dr. Brown was also responsible for the creation of over 40 patented small molecule compounds. The lead molecule has been demonstrated to reverse the effect of lethal doses of radiation in the GI tract. As a radiation mitigator and potentially a radiation protector, this compound has multiple potential applications for the military and civilian population alike. The lead molecule has recently been selected by 2 US government programs for testing. The second company from Georgetown University that is presenting today is Diviner Therapeutics. Diviner Therapeutics was founded in 2014 by Drs. Stephen Byers and Sivanathan Dakshinamurthy, based on Georgetown University's technology that relates to novel inhibitors of cadherin 11, a protein that is involved in cell-to-cell adhesion. Cadherin 11 inhibitors have potential therapeutic applications in high incidence cancers, inflammation, and autoimmune diseases. Diviner is currently optimizing and testing the lead molecule and has entered into a joint strategic R&D partnership with STQ Pharmaceuticals for advancing the compound for immunotherapy. In fact, Diviner is the only company with small molecule inhibitors against cadherin 11 and is therefore well positioned with a clinically validated target. It is actually worth to mention that Adheron Therapeutics with a cadherin 11 antibody asset was acquired by Roche for $580 million in 2015. Cancer Advances is the third company from Georgetown University that is presenting today. The company was founded in 2008 and is focused primarily on impacting human health and the progression of gastrointestinal cancers by enhancing the adaptive immune system. The company has a broad patent portfolio and is led by an experienced management team. The company has secured IP rights directed to clinical stage technologies from Georgetown University for the development of CCK receptor antagonists for the treatment of NASH and cancer. Also, the company has polyclonal antibody stimulator vaccine in clinical trials for the treatment of gastric, pancreatic, and colorectal cancer. The founders of the company are Dr. Alan Cato and Linda Sutton. Cancer Advances is a wholly owned subsidiary of Cato BioVentures. With this, I request Tokar Pharma to come and present.
Before we get started with our presentations, I'd like to thank George Washington University for organizing this event and for working so closely and collaboratively with Georgetown to create the first DC regional focused startup showcase. Thanks a lot, Steve.
Good day. My name is Damian Dolan, and I am the co-founder and CFO of Tokar Pharma, Inc. Tokar Pharma is the life sciences company focused on the development of novel industry-defining small molecule therapeutics aimed at countering threats to global public health. Today, the global public health problem we'll be discussing at length is that of increased exposure to ionizing radiation for both civilians and military personnel alike. The reasons for this increase are, one, the increased availability of radioactive materials over the past 50 years, and in particular, their storage in relatively unsecured locations such as universities, hospitals, and commercial settings. The increased potential for terrorist activity utilizing radiological materials, including domestically with the rise of local extremist groups in the US, the mishandling of nuclear waste such as hospital matter, natural disasters and their impact on nuclear facilities such as the Fukushima 2011 event, the side effects of radiation therapy, as well as less discussed radiation or space radiation once astronauts and individuals leave low Earth orbit. That being said, despite all of these areas that cause exposure to ionizing radiation, there are no FDA-approved treatments to counter radiation exposure at this time. The encouraging news, however, is Trokar Pharma's proprietary tissue platform technology has been proven to reverse cellular damage due to lethal doses of radiation. One of Trokar Pharma's patented lead compounds, YK4250, specifically treats GI acute radiation syndrome, which untreated has a high mortality rate with death occurring 9 to 14 days post-exposure. Non-clinical in vitro and in vivo studies have been conducted at 4 gray and 14 gray respectively. In all studies, initial treatment was provided a minimum of 20 hours and up to 72 hours post-exposure to ionizing radiation. Trokar's proprietary data demonstrates that YK4250 increases survival of mouse colonic epithelial cells. It also increases self-renewal of the same epithelial cells and increases intestinal crypt survival in mice. YK4250 and several other proprietary Trokar platform technologies mitigate the cytokine storm and have application for the treatment of chronic kidney disease, pulmonary lung fibrosis, immunotherapy boosting, and infectious diseases. The mechanism of action for YK4250 involves the renin-angiotensin system. Angiotensin II AT1 receptors in the GI tract, lung, kidney, and heart provide a major therapeutic target for targeting AT1 positive cells. YK4250 specifically targeted to angiotensin II AT1 receptors also inhibits reactive oxygen species, as YK4250 is a powerful antioxidant, and it also upregulates ACE2. The upregulation of ACE2 mitigates the cytokine storm as it has the beneficial effects of vasodilation and anti-proliferation. In addition, it is anti-inflammatory, anti-fibrotic, and anti-thrombotic. Our journey as Trokar started in 2009 when Dr. Milton Brown was approached by one of his fellow colleagues at Georgetown to develop a novel molecule that would inhibit reactive oxygen species. That again was in 2009, and that led to the creation of YK4250. Upon the creation of YK4250, successful non-clinical studies on hypertension were done in 2009 and 10, leading to our first composition and use patent being issued in 2016 after being initially filed in 2010. We also have additional composition radiation patents, as well as a second patent for another molecule as a radiation mitigator, which was received in 2019. After the 2010 study, there's a 2016 study on radiation exposure done by the University of Oklahoma in conjunction with Georgetown. In 2019, Trokar Pharma was formed, and in 2020, we are currently working with NIAID and AFRI, which is the Armed Forces Radiobiology Research Institute, to test separately YK4250 from a safety and efficacy perspective. When you consider the target market for Trokar's YK4250 GI-ARS mitigator, we consider both the military and the civilian population. The application for the two are slightly different. For the military, the expectation is that YK4250 would be issued as part of their standard issued gear, whereas for the civilian population, including the first responders, there will be stockpiles provided by the CDC as part of its strategic national stockpile program. This will be similar to what the CDC does for ChemPact in the case of chemical and biological exposures or incidences in the country. When you look at the market size for the mitigator, YK4250, we first look at the entire population for the US, Canada, Europe, and select Asian and Middle East allies to create our total addressable market, which we size at about 2.7 billion. We then consider, of that 2.7 billion, we take a haircut and say, let's consider 10% of the US population plus only the US military first responders and limit the numbers internationally to only the military, active military of US allies. And that leads us to about a 50 million SAM. Lastly, when you consider the actual market share for Trocar, we take a further haircut and we assume 10% of active US military and reserves, 10% of first responders in the US, 3% of US allies active military, again for the countries of Canada, 3 in Europe, which are France, Germany, Italy. We then also have India, Israel, Japan, Saudi Arabia, and South Korea. And that gives us a total market share or serviceable obtainable market of 3 million individuals. At a single-digit market share level, Trocar Pharma's initial projections would result in multi-billion-dollar annual revenues based on relatively conservative prices per dose of YK4250. The development pipeline for Trocar Pharma has been extensively created in conjunction with Trocar Pharma's advisors as well as third-party consultants. And it is extremely granular, and we will focus initially on the radiation mitigator, which is our primary target at this time. When you look at the radiation mitigator, as we discussed earlier, we are doing additional preclinical trials with NIAID and AFRI. The expectation is that we will be able to finalize our IND application process by the end of first quarter 2021, again, subject to financing, with the expectation that phase 1 will be able to start immediately thereafter and last 1 year, ending in first quarter 2022. Given the animal rule, the FDA's animal rule, the expectation will be that we'll also be able to conduct phase 2 surrogate studies using YK4250 simultaneous with our phase 1 clinical trials, which will result in our ability to move straight to commercialization of YK4250 by the second half of 2022. In addition, as we discussed earlier, we do have other therapeutics as part of our pipeline and as part of our patent suite. And the first one we'll discuss is there's a combo therapy we have developed for COVID-19 based on existing drugs in the clinic. On that basis, subject again to financing, we'll be able to start phase 1 clinical trials almost immediately. The second one is our novel therapeutic for COVID-19. We are currently doing preclinical testing with George Mason University. In addition, we have our immunotherapy boosting, CKD, and lung fibrosis therapeutics. The expectation is we will look further into those post-IND approval for our radiation mitigators, so the second half of 2021. Lastly, we have data for our hypertension drug, actually very positive data. However, due to the competitive environment for this product, as well as the long lead time to commercialization, we expect to only start actively pursuing that the first half of 2022, unless other parties are interested and we have funding that becomes available sooner. The development cost specifically for YK4250, we again, that is associated with our detailed development plan, is $24.5 million. Again, that's a total development cost. That is broken down into $2 million to finish our non-clinical efficacy studies, then another $5.5 million to get us through the IND process, and then $17 million for the combination of our phase 1 clinical trials and our phase 2 surrogate study. Lastly, in closing, I'd like to spend a minute to introduce the core leadership team of Trocar Pharma. We have as our co-founders, chairman and CEO, Dr. Milton Brown, who has a medical degree from the University of Virginia, as well as a PhD in organic chemistry. Dr. Brown also has 25 years of experience in drug discovery and has been a tenured professor at both UVA and Georgetown. And at Georgetown, he was the founding director of the Drug Development Program and endowed chair of experimental therapeutics. Dr. Skip Trump has over 40 years of experience in practicing oncology and is responsible for our clinical development and oncology program at Trokar. He was the CEO of Innova Shah Cancer Institute and was also president of Roswell Park. Again, I am Damien Dolland, and my background is investment banking and corporate finance. I was a managing director for JP Morgan, and I had spent over a decade at Morgan Stanley. And I've covered multiple products and industries, including healthcare and pharmaceuticals. On behalf of the Trokar Pharma team, we appreciate your time with us today. I look forward to further engaging with you. Thank you.
Hey, Damien. This is Charlie. Thank you for making time today. And the way I tend to ask questions, and I'll probably ask you three, one is sort of a softball to ease into things, and then usually a couple of questions either about adding value or clarifying something. So the softball question is, how did you choose the name of your company?
Well, the CEO, Milton Brown, is an avid fisherman, and the Trokar hook is a hook that has 3 points. It's a very sharp hook and precise. So that gave way to the name of our company. So we're sharp and to the point and precise in terms of going after drug discovery.
And investors. Excellent. Okay. All right. So then maybe the value add question. Have you guys thought about going after medical countermeasures priority review voucher?
I have not specifically discussed that yet at this time, so no, we have not.
Okay, I might suggest respectfully that you look into it. If you get through FDA and you were to get one of those, you can sell them and they still fetch a pretty good price in the market. Mm-hmm. So just maybe something to consider.
Thank you.
The second of the questions is, how do you plan to recruit for the radiation trial? It's not like, at least from my experience, you got folks walking around just saying, hey, I'm going to get a radiation burn in 6 months. Can I sign up now?
Yeah, perfect. That's a great question, Charlie. The similar way when they're doing drug discovery for Ebola and the like, we are using so for safety, yes, it will be done in humans as part of phase 1, but phase 2 will be done in a surrogate study using primates.
Okay, excellent. Because, yeah, I guess that's sort of ethical too then, right?
100%. That's right, FDA animal rule.
Okay. And last question, and then I'll pass it on to my other panelists. Have you guys been in discussions with BARDA at all about maybe a BARDA grant and about maybe at some point including your molecule in the national stockpile?
100%. So, for example, we had a conversation that was organized by NIAID. So it was NIAID, NIH, BARDA, NASA, and AFRI about 3 months ago. And at the back of that, NIAID has decided to take the lead to actually test our compound. So that's why we have 2 testings, one being done by AFRI in their HE model, and one being done by NIAID in their GI full body model.
Fantastic.
And so we're also still talking to NASA potentially as well. But yeah, so we've started that. Thank you.
Awesome. Sounds like you guys are firing on all cylinders.
Hey, Damien. I'll jump in quick. Sorry. Hey, Damien. Matthew Misa here. Great presentation. Just would like a bit more, if you could flesh out a bit more about the preclinical data you have so far, kind of validating the molecule in acute radiation syndrome. And are there any kind of key, almost pre-development experiments you need to run just to hammer that down further? Or do you have the data package ready to go and just ready to jump into IND enabling work?
Yes, thanks. Great question. And I wish Milton Brown was on.
I'm here.
Okay, Milton, maybe
I'm
you
here.
could join in as the creator and the scientist for this drug.
At the animal work has been done to look at GI-ARS in a standard model that's accepted. It was very potent in regards to keeping the animals alive. We use 14 gray radiation, which is a lethal dose. You don't expect any animals to live, but animals did live. 3 out of 4 animals lived more than 40 hours in the first study, and that they stopped the study at that time because that was the protocol. Longer term studies are being done, but at the moment, NIAID thinks that because of the 36-hour survival, that would be enough time to get you to a hospital to get a bone marrow transplant, which would be life-saving. So it's very, the GI syndrome is the first thing that kills individuals at 9 days. And so if we have a drug that overcomes that, it would make a world of difference in how we treat these types of patients, including our military and civilians.
No, absolutely. Thank you. That's really helpful. And on the pipeline in the slide that we saw, are those additional indications based on the same molecule or different molecules kind of related? Could you just flesh out that a bit more as well?
Yeah, we have several molecules. We have the lead molecule, mostly YK4250, has been lead in several indications, but we have come up with other molecules that actually would be backup molecules in case we run into some type of problem with any kind of problem. But the issue, YK4250 has multiple indications because of the utility of the AT1 receptor in regards to these different diseases.
Excellent. Thanks. That's all from me.
Thank you, Ben.
This is Peter. I have a question. I was wondering if you could elaborate a little bit on how this is applied, what the application is as far as delivery mechanisms, and what type of things might restrict its effectiveness, or what type of side effects it might have.
So this is Milton. I'll jump in, Damon. The delivery mechanism, it depends on the medical application. Sometimes with GI-ARS, and patients don't want to have oral administration, so you have to use a subcutaneous dosing because they're sick and they're vomiting and the likes. So we have we know our drug is orally available, has a half-life of 5.5 hours. We have also utilized it subcutaneously as well in our GI-ARS models, and shown its effectiveness by that route of administration. For toxicity, we haven't seen any toxicity up to 550 milligrams per kilogram. Our therapeutic dose right now is 10 mg per kilogram. So, I mean, we have a huge window of actually dose escalation as well as treatment. So we haven't really seen any overt. The only toxicities would be related to what the AT1 receptor, and that's a validated receptor. I mean, it has 10 drugs that are drugs for that target.
Yeah, guys, I think we kind of need to wrap up here. Sorry.
Well, thank you. Thank you, Charlie. Thank you to everyone.
Thank you.
Likewise. Thanks so much.
Hi, my name is Steve Byers. I'm presenting on behalf of Diviner Therapeutics and my partner, Shiva Dakshanamurthy. I'm a cancer biologist, and Shiva is a computational chemist and artificial intelligence computer scientist. And let me share my screen. So what I'm going to talk about today is one of the projects that Diviner has ongoing, which is called cadherin 11 inhibitors in cancer and fibrosis. And before I sort of leap into that, I just want to ask you a quick question. So what do these people have in common? It's a bit of a giveaway now since Ruth Bader Ginsburg just died, but the answer is, what is pancreatic cancer? And as many of you know, this is a lethal disease with a very poor 5-year survival. It's just about to become the third leading cause of cancer death. It's asymptomatic until really it's too late. Surgery is rarely curative. Alex Trebek seems to be doing okay right now, but generally it's not curative. And it's resistant to chemotherapy. Most chemos for pancreatic cancer are essentially palliative. It's resistant to personalized therapy, and it's resistant to modern immunotherapy. And this is sort of important because pancreatic cancer is classified as a cold tumor. And one of the reasons we think this is the case is that it's characterized by an extreme scarring of the pancreas, or a fibrotic reaction. It's not the only tumor, but it's the one that's the most fibrotic. And that's why we became interested in cadherin 11, as it's a molecule that's associated with fibrosis, all forms of fibrosis. These include heart fibrosis, rheumatoid arthritis, pulmonary fibrosis, skin sclerosis, inflammatory bowel disease. And we just wanted to see whether the fibrotic situation in the pancreas is in any way involving cadherin 11, and if so, if we ablate cadherin 11, does it improve outcomes? So first thing that you do is you look to see whether cadherin 11 is actually elevated in pancreatic cancer, and these are just box plots from the published databases, and it's dramatically increased, on average about 10 to 20-fold. And in keeping with its association with fibrosis, it's expressed predominantly by the cancer-associated fibroblasts. These are fibroblasts that are associated with the tumor, and in many cases in pancreatic cancer, the bulk of the tumor itself is made up of these fibroblasts, and you can see them here in that brown staining that's indicated by the arrows. The tumors where the asterisks are, and for the most part, they're negative for cadherin 11. Some human tumors are positive both in the fibroblasts and in the tumor cells themselves. So one way in which you prove the principle that your molecule of interest is associated with a disease is that you genetically manipulate it. And the way we do this is we take advantage of a mouse model, which is called the KPC mouse model, which is very much like the human disease. And we take that model and we ablate one or both copies of the cadherin 11 gene. And then we simply ask, do the animals that have 1, 2, or 0 copies of cadherin 11 survive longer or shorter than the control animals? And much to our surprise, we were anticipating we might see a change in fibrosis, but we weren't anticipating this result. So this is what's called a Kaplan-Meier plot. And on the left, you can see the black line. These are the control animals that have both copies of cadherin 11, and they survive about 145 days on average. If you treat those same animals with gemcitabine, that's the blue line, and gemcitabine is the standard of care, or one of the standard of care chemotherapies, it has very little effect. It really doesn't affect these animals at all. However, if you take away 1 or 2 copies of cadherin 11, so there's half as much cadherin 11 or no cadherin 11, you shift that curve significantly to the right. And what that means in human terms is this is a multi-year increase in survival. And to put that in perspective, if that were true for Ruth Bader Ginsburg, she'd still be alive today, and our politics would be significantly different. And if you look at the green line, this is what happens if you take away one allele of cadherin 11 and you add the standard of care chemotherapy. And what you see is that we now see a more dramatic increase in survival, and we begin to see actual cures. Some of these animals are actually cured of their disease. So in patients, we can't take away cadherin 11 genetically. We have to make drugs. But before I get to that, let me just summarize what we found so far. I'm not telling you, showing you all of this data, but basically taking away cadherin 11 converts pancreatic tumors from an immunosuppressive environment to one that is immunopermissive, and allows the endogenous immune system to now recognize the tumor. So in order to do this in a therapeutic situation, we need to develop drugs that will inhibit cadherin 11. And the way we did this was take advantage of Shiva's computational chemistry and computational abilities and design a small molecule drug that blocks the cell-to-cell adhesion domain of cadherin 11, and that's illustrated here. This is sort of important for us to develop a small molecule drug because this sort of region of a protein is not very susceptible to antibodies, so it's going to be difficult to raise antibodies against this. And lo and behold, if we treat animals with these inhibitors, what we see is basically a complete cessation of tumor growth. And we can see that in those two plots on the left. We give them a brief treatment at the beginning after we've inoculated with tumor. And during the time of treatment, it stops the tumor growth altogether. As we remove the therapy, the tumors start to grow, but they're still significantly smaller than the controls. And if we keep it there the whole time, we effectively stop tumor growth altogether. And the key experiment for us is to let tumors grow to a decent size and then begin the treatment, because this is much more like what happens in the human. We don't generally go into the human before the cancer is developed. We wait until it's there and then we begin treatment. And you can see in this bottom curve that if we let the tumors grow up to about 200 or 150 cubic millimeters, then begin treatment, we also significantly affect the growth of the tumors. So we're very excited about this. This is a potential for a treatment for pancreatic cancer and potentially other fibrotic disorders. And so our conclusions, I'm not going to go through all of these. This is pretty much what I've shown you. And what I'd like to basically get across is that targeting cadherin 11 by our drug is effectively a new mode of immunotherapy in pancreatic cancer. And my dogs are going berserk here, I'm sorry. We think the combination of cadherin 11 targeted therapy with a standard of care treatment, for example, chemo or some forms of immunotherapy, would actually result in cures. Pancreatic cancer isn't the only disease for which cadherin 11 inhibitor therapy is indicated. You can see the cancers here, and they're all bad players. The worst forms of breast cancer, the worst forms of prostate cancer, and brain tumors, and many other gastrointestinal malignancies have high levels of cadherin 11. So they're all potential targets for this therapy. In addition to that, I mentioned at the beginning that cadherin 11 is associated with fibrotic diseases, and there are many of them, and these are multi-billion-dollar markets. And each one of these diseases that's indicated with an asterisk is effectively cured in animals that have had their cadherin 11 genetically ablated. So we think the cadherin 11 inhibitor therapy has a very broad application. We have a robust patent portfolio, and our drug discovery pipeline is indicated here. We were formed in 2014, and in 2018, we licensed one of our patents from Georgetown and began to do the characterization experiments. We've completed phosphokinetics and phosphodynamic studies, which tell us that it's likely to be orally available. The experiments I've shown you so far are not oral experiments, they're injected, but we think it's likely to be orally available. What we'd like right now is the next step for us is to seek what's called investigational new drug status, IND. And in order to do that, we've already invested somewhere in the order of $4 to $6 million, which includes money to the company and money from grants that we used before that. We need another between $1 and $3 million to get IND status. That we should anticipate would occur within the next 6 months to 8 months. And we'd like to be able to complete our phase 1 studies in-house. That's a much more expensive proposition and will cost us about $10 million. The reason for that is if you do the studies in-house and now you go and you partner and look for partners in big pharma, the value of your company is increased about an order of magnitude. So that's what we're here for. We're here to present this to you and see if there's any interest. Thank you very much.
Hi, Stephen. This is Niall. Thanks for an
Hello.
excellent presentation. I thought you spoke with great clarity, something even I could follow. So I wanted to ask some questions, kind of your plans going forward. My understanding is that your immediate need is to raise $3 million, right?
We're sort of halfway through studies that we think would allow us to apply for IND in the next 6 months. So we spent about $400,000 from the company, and we have a lot of sorry, that's my phone going on. And we have a lot of support that we've got from grant applications. So we need about another between $1 and $3 million to complete IND studies.
Okay, and do you have a ballpark of how much you're going to need to raise before you can get to exit?
Well, that's one of the reasons that we're here. We would prefer to carry out the phase 1 studies in-house, or at least partnering with people that before we exit. And our partner right now would prefer to exit before we do the phase 1 studies. The reason for that is, you're a business person more than I am, but I think the value of the company is dramatically more if you've already completed the phase 1 trials.
Okay, thank you. And what are your thoughts with regard to your team? I mean, as you move forward, I know you and your partner both have sort of strong scientific backgrounds. Would you consider expanding your team to bring in business or commercial skills?
Sure. We've had somebody that helped us with the licensing applications and was a beginning CEO, and we'd be looking for somebody that's working to the next level, somewhere between $10 and $100 million of investment. As I said, we partnered. We have a revenue sharing relationship with a company that's supported us. They have 30% revenue share in the small molecule, and we're working with them also to develop a high affinity antibody, for which they have the predominant share.
Great. Thank you very much.
Great. Thanks, Stephen. That's a great presentation. So just a quick question on the second generation compound. What optimizations did you do as a drug-like as it stands? And do you have composition of matter IP being filed on the second generation?
On the second generation? We have not filed patents on the second generation yet. We're sort of trying to figure out whether we should do that through Georgetown and license it, or whether we should do it through the company itself. That's basically where we're at. The second generation compounds were based on the PK/PD studies that we had with the first generation, which were pretty encouraging, but it allowed us to increase the efficacy in vitro by about an order of magnitude with the second generation compounds, but they have not yet been patented.
Great, thank you.
Okay, if there's no more questions, then Stephen, thank you so much. Great presentation, really promising molecule, worthwhile disease. Thank you very much.
Okay, thank you.
Good afternoon. I'm Dr. Jill Smith from Georgetown University, and it's my pleasure to present to you this afternoon some of our research involving developing the CCK receptor antagonist proglumide for NASH and pancreatic cancer. NASH is a condition of high unmet need. NASH refers to non-alcoholic steatohepatitis, which is a type of non-alcoholic fatty liver disease. In the United States, NASH has become a pandemic with about 15 million subjects involved and affecting 5% of the United States adults. The histologic hallmarks of NASH include liver cell damage, inflammation, fat in the liver or steatosis, and fibrosis. There are few or no symptoms of NASH. The risk factors include obesity, metabolic syndrome, type 2 diabetes, and hyperlipidemia. The diagnosis involves medical history, blood tests, such as elevated liver enzymes, imaging tests, such as ultrasound of the liver, and sometimes a percutaneous liver biopsy. Currently, there are no medications approved to treat NASH or fatty liver disease, and the only therapy that works is weight loss. One problem with NASH is that fibrosis or scar tissue, which occurs in NASH, increases the risk of hepatocellular carcinoma, and this is the fastest increasing cancer in the United States today. Also, another disease that involves fibrosis is pancreatic cancer. Pancreatic cancer is a complex disease with a current 5-year survival rate of only 9.3%, which is the lowest of any cancer. Unfortunately, at the time of diagnosis, 90% of those with pancreatic cancer have unresectable disease or metastatic disease. There is a multidisciplinary approach to this disease, including surgery when possible, chemotherapy with or without radiation. However, chemotherapy with the standard of care, which is Gem-Abraxane or FOLFIRINOX, does not improve survival more than 1 year. The poor survival rate, in part, is due to the dense fibrotic microenvironment that inhibits the chemotherapy drug penetration into the pancreatic tumor tissue. Pancreatic cancer is also highly resistant to immunotherapy because of the poor T-cell infiltration and high fibrosis, also the low mutational burden. Currently, the therapy used for pancreatic cancer also is not target specific. Pancreatic cancer is increasing in incidence, and this year, it's about 56 to 58,000 will be diagnosed with pancreatic cancer, and most will succumb to the disease. This year, pancreatic cancer has surpassed breast cancer in cancer deaths and has now become the second leading cause of cancer mortality in the United States. And by the year 2030, approximately 65,000 will succumb to this disease. The global pancreatic cancer therapeutics market is projected to grow up to about $3.5 billion in the year 2023. Some of our background data is shown on this slide for the preclinical studies that were done using the CCK receptor antagonist proguanide in liver disease. In NASH, on the top left-hand side, you can see a liver histology stained with Mason's trichrome stain, and it shows the characteristic features of NASH, including steatosis, fibrosis, which is the blue stain here, and inflammation. However, in a mouse that was also treated with proguanide and had the same high-fat diet, there are none of those features found. We found in our preclinical trials that proguanide reversed all of the histologic features, including inflammation, fibrosis, and steatosis. It normalized the liver enzymes, and the most important finding in our study was that we found that proguanide prevented the development of liver cancer. In pancreatic cancer, the CCK receptor becomes expressed on precancerous epithelial cells and is markedly overexpressed in pancreatic cancer. We also found that the CCK receptor is overexpressed on fibroblasts or stellate cells, which are the cells that are responsible for causing the fibrosis in the tumor microenvironment. In a mouse that was treated with proguanide for 4 months, compared to a mouse control pancreas, you can see the extensive fibrosis stained with blue on the left compared to the decreased fibrosis and the decreased cancer epithelial cells in the proguanide-treated mouse pancreas. The other important finding, besides decreasing cancer epithelial cells and decreasing pancreatic tumor fibrosis, is that we found that the mice treated with proguanide had an influx of CD8 positive tumor infiltrating lymphocytes into the tumor, which is one of the reasons that we found that this therapy enhanced the efficacy of immune checkpoint antibodies. Proguanide is a non-selective CCK receptor antagonist that was initially developed to treat peptic ulcer disease. It exerts its inhibitory effect on gastric acid and reduces gastric motility. However, development for an antacid drug, this was stopped back when the proton pump inhibitors were developed. The mechanism of action for programmide includes that it blocks both the CCK-A and the CCK-B receptors. It has been shown that it is orally bioavailable and has been safely used in doses up to 1,600 milligrams per day. It has a broad safety profile, and the method of use intellectual property patents have been filed using this drug as an antifibrotic agent, both in pancreatic cancer, in combination with immune checkpoint antibodies or standard chemotherapy, and also as an antifibrotic agent in reducing NASH and reversing NASH and preventing liver cancer. The current status of our development includes that we had a publication in 2018 showing the combination effect of programmide with immune checkpoint antibodies. The second publication was showing that programmide halted non-alcoholic steatohepatitis and prevented hepatocellular carcinoma, which was published this year. The method of use patents have been filed on both of these discoveries. In addition, we have been granted orphan drug designation for the use of programmide in pancreatic cancer. Currently, at Georgetown University, we are conducting a phase 1 study to test the safety and tolerability of oral programmide in subjects with NASH. This study is funded by the NIH, and the clinicaltrials.gov website number is shown on your slide. We also have approval from the FDA for a phase 2 trial with pancreatic cancer in combination with standard of care with programmide, and we're hoping to move this into a clinical trial in the next year. This is the timeline frame that we're talking about for developing programmide, and on the top you can see the development timeline for liver disease and NASH. We're hoping to also develop this for a treatment as an antifibrotic, such as those who might have cirrhosis. And the pancreatic cancer development timeline is shown on the bottom of the slide. The Cancer Advances team is shown on this slide, and you can see Dr. Alan Cato, who's the CEO, and Linda Sutton, who is the president of Cancer Advances, and the broad experience that comes from this team. The phone number is shown for Katie, who is down at the bottom, and my email address for anyone who might have scientific questions concerning the development and this drug. Programmide has the potential to be an important therapeutic in both of these areas because of its antifibrotic effects. In NASH, there currently are no approved therapies. There is potential to use programmide by itself or combination with other drugs that may be tested on the market. For pancreatic cancer, currently there are very few options, and there is a gap in our treatment of this disease. Desperately, there are new drugs are needed to treat pancreatic cancer. One advantage of programmide is that not only does it inhibit growth of pancreatic cancer cells, but it also blocks the fibrosis that impedes the penetration of chemotherapy or immune checkpoint therapy or immune cells itself. The use of the funds is shown on this slide, and we hope that with your support that we can move this product into the clinic and get approval, an early approval from the FDA for these two desperately needed diseases that need new therapy. Thank you for your attention, and we're happy to address any questions.
Hey, Katie, thank you for kind of walking us through that. Impressive work, worthwhile diseases to tackle. I'm going to ask you three very quick questions that are all interrelated to each other. So the first is, let's assume that you get your method of use patent or method of treatment patent. if this is an FDA approved molecule, and if there are, for example, generics out there, how do you differentiate yourself in such a way that a doctor doesn't look at it and say, you know what, I'm just going to write a script for the generic, give the generic to the patient, and bypass sort of the higher priced exclusive brand?
Yeah, so this is a question we also thought about since proguanide is an older drug. And we discovered that proguanide was never approved in the US. So at minimum, we could get new chemical entity, perhaps. We've also filed for orphan drug exclusivity and received it for pancreatic cancer, and could potentially do so for liver cancer prevention. Another potential way to differentiate is, depending on the clinical program, to develop a formulation that might work better, as perhaps in the NASH indication. If it's a slow release, then it wouldn't be able to be easily replicated by a generic. But right now, research has shown there really aren't generics available in the US, as this drug was never put on the market in the US.
Fantastic. Okay. Good answer. Thank you. So then the next question is sort of when you're talking about NASH, right? People who, the big fear among NASH people, obviously you don't want to develop cirrhosis, you don't want to develop liver cancer, but people really are concerned about what they call decomposition day, right? It's when your liver stops working well enough that you end up in the hospital, and then from that point on, you sort of continuously have problems. How would you guys run a human clinical trial against NASH? What would for example, your primary endpoint in phase 2 be?
Well, that's a really good question, and something we're talking to Jill about, and looking also at some peer data. I know that the FDA requires that you have to either show one-step improvement on, I think, the steatohepatitis score, or improvement of the fibrosis. So what I believe we would do is try to, and they also usually require liver biopsy. So, which isn't a big help when you're trying to enroll patients. No, people don't really want to do liver biopsy. So what Jill and we have talked about is defining markers that, histological markers that would show an improvement. And that's what I believe a couple of other companies have done, like Kiro and Intercept. They sort of developed their own like validated test. And unfortunately, Intercept didn't then get approved after that. But that's what we were thinking about, trying to have the initial take, measuring the histological markers, and then you could do the follow-up study measuring, I guess, the clinical benefit, where you would actually, show the liver biopsy.
Okay, thank you. And then last really quick question, because you mentioned that you might be coming to market sooner through the accelerated approval pathway. Have you guys looked at the patent landscape? Are you comfortable that you have freedom to operate?
We are doing an extensive review of the IP right now, but that's one area that we believe that should be clear, at least in the US.
Thank you.
Thanks. Hi, Katie.
Hi.
I concur, great presentation. Thanks for that. I just have one question, sort of, in your go forward plan, I mean, pretty basically it was to seek clearance for both indications, so do like both sets of trials. Could you maybe talk around some of the considerations why that's the preferred strategy, or we're picking one indication and trying to get it cleared first?
Yeah, that's a good point. And I guess it also depends on being able to get the investment to go parallel track on both studies. It somewhat depends on funding, but to be able to go after both indications. An alternative would be to potentially go after the smaller oncological indications first, and because those would be smaller studies, and guarantee the orphan drug designation as well, and then perhaps then go after a larger indication once we have some revenue generated.
Katie, thank you. I'm sorry I need to interrupt because we're out of time. But thank you for a very impressive presentation. And we're looking forward to seeing all the good results that the company generates going forward.
Thank you.
Thank you.
Let me first take a couple of moments to tell you about our Technology Commercialization Office. Our TCO was re-engineered about 7 years ago with the goal of better connecting to the outside business and entrepreneurial world, with a focus on speed of execution to better serve our faculty inventors. All the traditional metrics have more than doubled, and our licensing income has increased more than 50-fold, making us the fastest growing office in the United States. These results are a direct result of our great, talented team. It's always about the people. Our first startup is LuxMed Systems, with a therapeutic device for cardiac arrhythmias, utilizing optical sensors for rapid and effective cardiac ablation treatments. The CEO is Omar Amirana, and the GW inventors include Dr. McCander, Sarvasian, and Kay. Our second startup is Advate, a biopharmaceutical company developing novel treatments for debilitating diseases. The GW technology is a therapeutic for melanoma skin cancers. The CEO is Karthik Musunuri, and the GW lead inventor is Dr. Balagra. Our final startup is Nanocon, developing a novel implant to repair joints in younger active adults who are not candidates for the traditional joint replacement treatments. The CEO is Dr. Ben Holmes, and the GW inventors include Professor Grace Zhang and Drs. Holmes and Castro. Enjoy the presentations.
Hello, and thank you for joining our presentation. My name is Karthik, and I'm the CEO for Advite, a biotechnology company that is dedicated to developing novel diagnostics and therapeutics to help patients suffering from a variety of debilitating diseases. We currently have around 15 employees and are headquartered in Malvern, Pennsylvania, and also have a CLIA high complexity lab based out of Chicago, Illinois. Just a forward-looking statement. And the purpose for my presentation today is to introduce you to one of our very exciting programs in the oncology space, which is ADV300. ADV300 is a novel small molecule HDAC6 inhibitor that we plan to use in combination with PD-1 checkpoint inhibitors for the treatment of stage 2B through 4 melanoma. This special molecule downregulates PD-L1 expression on cancer cells and can increase the overall response rate. Currently, the problem is that only 20 to 30% of patients that are on anti-PD-1 immunotherapies actually respond. So there's a significant unmet medical need in improving the overall response rate. At Advite, we hope to use these highly specific HDAC6 inhibitors in combination with checkpoint inhibitors to increase the overall response rate and provide yet another effective and very safe therapeutic for these patients. In regards to the opportunity is there. Primarily, we hope to focus on the patients that have already developed some resistance to the checkpoint inhibitors, and also use it as an adjuvant therapy to existing therapies that these late-stage melanoma patients are already on. Our primary regulatory pathways would be to pursue ODDs for a number of indications, including stage 2B through 4 melanoma. Overall, in the US, there's around 90,000 patients that are diagnosed with melanoma each year, and around 13,000 of those develop resistance to these checkpoint inhibitors. So there is a huge unmet need with tens of thousands of patients that require additional therapy for which is responsive for them. The overall deal flow with HDACs in specific and the overarching epigenetic modifier field, it has been ever growing since the early 2000s. There's big deals around HDACs for primarily pan-HDACs, which target all the HDACs, but the M&A structures are there, and anything regarding novel approaches to tackling immunotherapy resistance has seen similar deal flow. Now to the product itself. Our ADV300 compound selectively inhibits HDAC6 while avoiding the side effects of pan-HDAC inhibitors. The compound is Ames negative, so there's no mutagenicity, and it is a small molecule. This compound is also tumor agnostic, so it could theoretically target any tumor type where current PD-1 inhibitors are used. These include different lymphomas, breast cancer, and other types of melanoma. In the below model, you could see that our compound has been proven in vivo models on syngeneic mice, showing significant tumor growth reduction. And why is HDAC6 a very important target? Why do we target just HDAC6? Well, to provide you some background, it all starts with the PD-1 pathway. When PD-L1 is expressed, this is a very important co-stimulatory molecule that's actually expressed on cancer cells, and this activates the regulatory pathway PD-1 in T cells. Usually these checkpoint inhibitors such as Keytruda and Opdivo target this PD-1 receptor on the T cells. And one of the necessary important events for activation of the PD-L1, which has to bind to PD-1, is the recruitment of STAT3, or the phosphorylation of STAT3. This is necessary, or one of the key ingredients that's necessary for STAT3 to activate is HDAC6. So HDAC6 is involved in numerous countless processes, including STAT3, which is highlighted on the right side of the screen. As you can see in these models, ADV300, also noted as SS208 in the graphs, decrease expression of PD-L1 in melanoma cells. It decreases the STAT3 activation that is measured in the left western blot. In the middle screen, you can also see that only HDAC6 inhibitors actually downregulate PD-L1 gene expression by reducing STAT3 activation. In pan-HDAC inhibitors, as is seen in the graph C and D, they actually upregulate PD-L1 gene expression. So this is a significant difference for why we target HDAC6 in particular. And again, on the right side, we have in vivo data to show that decreased tumor growth compared to just the control. This is also another reason why this is very synergistic to use with existing immunotherapies, especially anti-PD-1 treatments, because we are able to not just inhibit PD-1 on the actual cells itself, we can also inhibit or downregulate PD-L1 on the cancer cell. So we are indirectly de-promoting that activation. And the other part of this is how we indicate effective anti-tumor immunity in the tumor microenvironment. And this is by looking at the M1 to M2 macrophage ratio. A high M1 to M2 ratio indicates better prognosis for most patients. And we have shown that with ADV300, we do see an increase in the M1 to M2 ratio as seen in the tumor microenvironment. This is a very significant and unique finding for specifically highly specific HDAC6 inhibitors. In regards to our strategy is to target as many orphans as we can within cutaneous malignancies, including multiple lymphomas, melanomas, all across around in that space, and to expand the label once we have positive readouts from phase 2 clinical trials. We will also explore corporate partnerships and other out-licensing opportunities once we get to that stage. Again, I want to reemphasize that ADV300 is tumor agnostic. It can be applied to virtually any tumor microenvironment where current checkpoint inhibitors are used and where there's a high M2 to M1 ratio, because we have shown that we can actually improve that ratio in the tumor microenvironment. Currently, we're raising a seed round of $6 million that will be used to fund the IND enabling work that will enable us to get into the clinic by hopefully the end of next year or going into 2022. We will use this for preparing that IND and also any other internal R&D expenditures. And this just lays out some of our key upcoming milestones that we hope to accomplish. We'll definitely be filing an orphan drug designation for the appropriate target indications which we are pursuing by the end of this year. And we will also have a pre-IND meeting with FDA, hopefully by Q1 of next year, which is 2021. And on the screen, you can see our team. Obviously, I could not fit everyone that's a part of Advite on here, but we do have around 15 employees. We have a very brand new state-of-the-art R&D facility in Malvern that enables us to plan for the development of any small molecule from the preclinical stage all the way through the clinic, everything excluding in vivo work. We also do have a high complexity CLIA laboratory in Chicago, Illinois, that's equipped to do high throughput testing. It could also test different tissue types, and we can do a lot of biomarker research there in specific relation in the oncology space. Most of our team comes from very large and reputable pharmaceutical companies, as well as esteemed academia institutions. We all have many publications and a very great reputation and fixation for trying to improve human health and provide for better outcomes for patients. This concludes my presentation. If you have any more questions or wish to learn more about Advite, you can also visit our website at www.advite.com.
Hey, Karthik, thank you for that really interesting presentation. And your lead molecule looks especially intriguing, right? No like bushel basket of chiral centers that you have to deal with in synthesis. It looks to be a pretty stable aromatic ring and a heteroaromatic ring, amide bond. But one thing that kind of stands out that you don't see in a lot of drugs are two chlorine atoms on the aromatic ring. Can you guys, to the extent you can, to the extent you've tested it, do you anticipate that that might cause any problems when you give it to humans?
No, I don't think so, mostly because we've looked at a lot of other, even the pan-HDACs, which in some cases are not as favorable in terms of toxicology and safety. We've seen those get entered into humans, and some of which have ultimately gotten approved, and similar structures as well, but they also were able to succeed and go through the clinic very seamlessly. So I think that with the profile of this particular molecule and the composition, that we feel very strong that it's favorable when transitioning to humans. Of course, we have to prove that.
Okay, well, I mean, everybody does, right? It's part of the game.
Yes.
Okay, one other just very quick question, and then I'll pass it along. Have you guys run the compound against, for example, human liver microsomes and identified, do you have like a primary metabolite?
No, that's a good question. So we don't have one as of yet, but we do hope to conduct a lot of those models and during the IND enabling work as well, so that we could identify certain markers that may help us when we transition to humans.
Yeah, it'd be really interesting just to see, right, like where metabolism occurs, especially with those two chlorine atoms on the ring. I
Yes.
think it might sort of, if enzymes were freakable, it might sort of freak out some of those liver enzymes. So that's cool. Thank you.
Thank you.
Thanks, Karthik. Yeah, great presentation. I'd love to get your thoughts on digging a bit more in how selective the molecule is, and specifically how this can be a differentiator, given that there are other selective HDAC6 programs. You know, Keris has one in phase 1 trials. If they have a similar profile and are a bit ahead of you, kind of how do you compete?
Sure. So I think that while there are other HDAC programs, again, ours is Ames negative. A lot of the other programs that we've seen published are Ames positive. So when you have mutagenicity and you're translating into humans, there can be other factors that play into role. And it's also a reason why in the past, some of these programs have failed ultimately. I think the safety profile that we've looked at for this particular molecule is definitely favorable, not just compared against the pan-HDACs, but other selective HDAC6 inhibitors as well. In terms of the response, we still do see a very good signal, at least in vivo, again with ours, against other selective HDAC6 inhibitors. And I think just down to the mechanism, just down to the synergy it has with the combinations of other checkpoints, we feel very confident that this particularly program, if, of course, there's always going to be competition, but our specific approach and the specific disease we're going after is pretty unique. So I think that we try to, as long as we're ahead of the game, that'll be our entrance into the market.
Great, thank you.
Karthik, I have a question sort of around your company. Maybe give us a little context.
Sure.
Like I see that you have 15 employees, you've 2 facilities. It suggests you've been around a while and made some progress, but more specifically, could you talk about how long you've been around, how much has been invested, and
Sure.
you know, how many of these people work full time? I want to get a sense of what the company looks like today.
Sure. Yeah. Sure. So definitely, we were actually formed out of UIC, University of Illinois at Chicago, in 2017. So we spun out of there with some IP from my co-founder's laboratory. Dr. Jain is a very famous corneal surgeon, but he also has a knack because he also treats cancer patients who have ocular graft versus host disease. So that was a very complex disease, but we developed some IP around that in terms of looking at different biomarkers and also on some repurposed drugs. And throughout the past few years, we've been primarily R&D, but we did receive large amounts of non-dilutive financing, upwards of $10 million in the past year alone, to sort of help on the not just COVID front, but also looking at novel diagnostic approaches as well. So we have a team of around 13 full-time employees, so molecular biologists, immunologists, almost every specialty that are dedicated to innovating novel diagnostic products, and also a lot of them have expertise in the oncology space, especially with small molecule development. So we do hope to utilize our existing capabilities with, we have a new like FACS, FPLC, HPLC. So we have a lot of good specialized capabilities to assist with the small molecule development besides other programs.
Perfect, thank you. Sorry, we kind of have to wrap things up, but it really is a nice looking small molecule. And I think all the panelists are very excited when you guys go into the clinic to see kind of what the results you get are.
Yes.
So looking forward to it.
Thank you. Thank you.
Hello, I'm Omar Amarana, President and CEO of LuxMed Systems. I'm here to share with you why LuxMed is a terrific investment opportunity and we believe we're going to transform arrhythmia therapy and cardiac ablation. Notably, we like this image here on the right because, like these rays of light penetrating through the water, we're using light to penetrate into heart tissue to identify what's going on during a cardiac ablation. And this has clinical value, is extremely helpful and unique in the marketplace. Our investment opportunity hinges on a number of solid attributes. Specifically, we're solving a major problem. With these rays of light that interrogate cardiac tissue, we're able to identify remnants of tissue that were missed during the ablation. And we believe this will become important in becoming a first line of therapy. We're in a market that is highly underpenetrated, but is a multi-billion-dollar market growing at over 10% a year. We've got all of the key foundational elements in place to execute and to achieve regulatory approvals in order to commercialize on this Series A. We have validating human data. We're close to a design freeze. We've got very attractive reimbursement in place. We have development partners that we're currently working with who are experts. We've got a patent portfolio of 21 issued patents. Our regulatory path has been vetted by the FDA already, and it's very well known and established how we will get our first approval and then expand our labels. We've got a team that is steeped in electrophysiology and cardiology experience. Our cap table, interestingly, because this is a Series A, is completely clean. We have no institutional money in our deal. And that makes us a very attractive near-term risk-adjusted ROI opportunity. Notably, $12 million buys us 2 product approvals, one in Europe, one in the US, and we believe that puts us in a position for a possible exit. Exits in this space have been remarkable, averaging somewhere in the $250 to $350 million range. So this would represent over a 10x return in under 24 months. The market for treating cardiac arrhythmias is staggeringly large. There's 30 to 40 million patients with atrial fibrillation alone. There are over 1 million patients that get ablated a year, and this is growing at approximately 10% a year. 400,000 of those, or approximately 40% of cardiac ablations today, are atrial fibrillation patients. Notably, our catheter, the OmniView, would ablate all arrhythmias. So we're not limited to one portion of the market. What we do is capitalize on the existing procedure and the existing workflow. We've integrated optics into an ablation catheter that would be used seamlessly in today's procedure. Today, a catheter is introduced into the heart, placed on the location that's causing the arrhythmia. We basically do this procedure today in a blind fashion, and basically we're introducing optics into this procedure to interrogate the heart wall such that we are identifying what is behind or inside the wall in order to understand if we've achieved our objective to ablate all of the tissue. Very similar to a stud finder when you're trying to hang a picture on a wall. If you don't know where the stud is, you're really effectively putting a nail in that wall blindly. This is our catheter in a schematic, and basically these are optical ports. So at any given angle of attack, this electrode couples light into the tissue. Interestingly, we get an optical signature back from the tissue as soon as the electrode is touching the tissue. The blood inside the heart gives us no signal back, so we know if we're touching the tissue or not. Interestingly, we are using this optics to identify if the tissue is alive or if the tissue is dead. And we do this by exciting a specific endogenous molecule, NADH, in the tissue. If it's ablated tissue, NADH is no longer present. Here's an example of our catheter going over or being dragged across a segment of freshly excised but still viable tissue. And you see two white dots or ablation sites. These are tissue sites that have been ablated and are therefore dead. And you see in the blue column the LuxMed signal being very low, while over those two ablation points or ablation spots. But in between those, there's a gap or a segment of viable tissue, and you see our signal spike. This is a key differentiator of our system from all other ablation systems. We've tested our system in patients already, even though we're a seed stage funded company. And we've done 11 patients with 100% success that validates our system's operation. We have terrific economics, which creates value for all stakeholders. The procedure itself is reimbursed at $18,000 to $21,000 to the hospital, approximately $1,200 to the cardiologist or electrophysiologist. Our catheter costs us less than $500 to manufacture, and we would sell it at approximately $2,500, which is less than today's standard ablation catheter. That would afford us an 80% gross margin. We believe we'll have phenomenal yields in excess of 90%. That affords the hospital significant cost savings, opportunity for improved revenue per lab, and that basically would benefit the physicians and patients through better, faster, safer procedures, and obviously would benefit payers. From a milestone perspective, the two things to note are with a Series A, we will achieve a CE mark and an FDA approval on our ablation catheter. This would be a PMA submission. We expect this money to last over 24 months, and that would set us up for optionality around an M&A event or a Series B financing with an approved product in hand. A Series B has been modeled out in detail. A $35 million Series B would take us beyond profitability. So this would be the last capital in, would allow us to expand our labeling with the existing system, and would allow us to introduce a second energy source, which is today new and investigational, but quite exciting. We also would introduce predictive analytics and algorithms to optimize outcomes. Our existing FDA path to approval is both short and inexpensive, and our protocol and trial design would mirror the trial design that Endosense took before it was acquired by St. Jude Medical, which is now merged with Abbott or acquired by Abbott. They achieved approval on 43 patients. We're budgeting 60 for the CE mark and up to 150 for the FDA trial. We think that there's a continuous stream of value creation based on label expansion, market expansion, technology expansion, and the ability to introduce predictive analytics. One area for our pipeline is in an area called PFA, or pulsed field ablation. This is the new energy source that I mentioned, which is still investigational. Interestingly, even though we're only seed stage funded, we've already generated feasibility data here indicating that our optics is compatible with PFA and registers tissue changes in real time due to the PFA impact on the tissue. And we've got issued patents that cover PFA ablation catheters that are optically enabled. Our data analytics or predictive analytics opportunity is largely born out of these optical signatures that we will be capturing before and after ablation. And that will allow us to create algorithms based on a repository in the cloud of optical signatures that can help the physician go back in real time during a primary procedure using our catheter and interrogate and then re-ablate, if necessary, a given spot that might need touching up to prevent recurrences in the future. Our financial plan goes out through 2027, and two things to note. One is that we would begin our Series B discussions next year along with M&A opportunities. We would launch early in 2022, and that would afford us a revenue growth of 3 years to profitability on approximately $28 to $30 million in 2024. The competition is significant, but that's because this is an excellent market and is incredibly underpenetrated and very large with excellent reimbursement. The market essentially started with RF ablation and temperature sensing only, then incorporated force sensing technology to tell cardiologists how hard they're pressing on the tissue. But we believe ultimately you need to be interrogating the heart wall during ablation to really truly understand if you're doing a good job and to prevent recurrences in real time. The exits in this space have been significant, ranging from $250 to $350 million. And this list over the last decade of exits demonstrates that the last 4 exits in the space have all been pre-revenue exits. So we believe there's ample precedent that an M&A event could occur on our Series A achievement of regulatory approvals for our catheter in under 24 months, representing a 10x investment opportunity from our Series A. Our team is steeped in EP and startup and commercialization experience. I've been in electrophysiology since I was an engineer right out of medical school in Silicon Valley in 1992. I worked on the first RF ablation catheter that was FDA approved. I was formerly the VP of Business Development at St. Jude in their ablation division, which was doing approximately $500 million in revenue at the time. I'm on the board of several EP companies. I've been in electrophysiology for close to 3 decades. Terry Ransbury, our business partner and CTO and board member, co-founder, is a Duke biomedical engineer. He's been in over 600 ablations when he worked for Biosense Webster J&J and has been in a number of EP related companies. John McCarthy is a co-founder and board member, and he was the former CFO of Exact Sciences, having taken them public, and has been in numerous startups. AJ or Arun Jagannathan is formerly from Boston Scientific's business development and marketing, and has a venture capital management consulting background. Rick Randall is a board member and is a serial entrepreneur, former CEO of Target Therapeutics. John Fletcher is the former chairman of Spectranetics, having been instrumental in the sale of Spectranetics to Philips for over $3 billion. Dan Levangie is a serial entrepreneur, has generated terrific returns for his investors, and he's an advisor. Our SAB is world-class and includes Jeremy Ruskin, Vivek Reddy, David Haynes, and Andrea Natali. They are all key opinion leaders in this space. And as a result of all of these accomplishments and the team we have in place, we believe this is a compelling investment opportunity that solves a major problem, addresses an enormous underpenetrated multi-billion-dollar growing market, has all of the key foundational elements in place to achieve approvals on this Series A and $12 million raise. We have an excellent EP savvy team. We've got a completely clean cap table. And interestingly, and recently, we have received terms from a lead prospective investor. So we are now looking to syndicate on this term sheet And we believe this Series A would represent, excuse me, a fabulous investment opportunity for anybody interested in impacting the cardiac ablation market. And with that, I thank you for your attention.
Thank you, Omar. This is Peter Weissman. I appreciate your presentation. I think this is a very exciting device. I did have a question. I was curious as to any limitations on the utility or the effectiveness of the device. So for instance, as far as the target tissue goes, is there a minimum size, either the depth of the tissue or the width of the tissue that's being detected that you're able to detect? And with respect to that, what percentage of ablations are in that target range?
Great question, Peter, and you're getting right to the heart of something we've spent some time understanding and are pursuing. We have initial data that shows we have the ability to measure lesion depths beyond 8 millimeters. The typical atrial wall thickness, especially around the mouth of the pulmonary vein, happens to be in the 2 to 4 millimeter thickness arena. We're working with an optical development house that happens to have invested in us cash, and they're specifically working on this very topic. So we know that this is important. Our first label will not have lesion depth, but we aim to have lesion depth further down the line with the appropriate data in hand.
Thank you. And when this is being done, is this change the medical procedure at all, or is it the standard type of ablation? Is it open heart? Is it less invasive?
Every single physician trained in ablation today uses a catheter like this and is already trained on this particular catheter. This is called a point source RF catheter. We specifically chose to embed George Washington University's optical technology that we licensed into such a form factor specifically because it will not change the procedure dynamics or any of the workflow. Rather, it will make the workflow faster, saving time, reducing the number of ablations, potentially reducing fluoroscopy exposure, but most importantly, over time, demonstrating to the clinician that we are providing insight as to what they're doing, which is currently not available today.
Okay, thank you very much. I think we're out of time, but again, very exciting technology.
Thank you.
Hello, everyone. My name is Dr. Benjamin Holmes. I'm the founder and CEO of NanoCon. We're here today because we have an open round with roughly a third of the round committed and are looking for a lead investor. And I'm going to tell you how we're innovating the sports medicine space and knee rehabilitation today. So the problem that we are addressing is joint defects, specifically looking at damage to the surface cartilage in the knee, more commonly known as osteoarthritis or a sports injury. Now, there is already a very tried and true treatment for patients suffering this type of damage, and that's a total joint replacement. However, joint replacement surgery is very invasive, removes a lot of healthy tissue, and then also does have a long but limited lifespan. And thus, it's really only an option for older patients. And so we're really focusing on younger and more active people, which sustain this type of knee injury and need a surgical intervention, but just don't really have the same sort of like good go-to option that general orthopedic patients have. And so that's where we really see NanoCon fitting in. We're developing a new type of implantable medical device for treating this type of cartilage damage. It's essentially a patch, an implant with a relatively large surface area, but the thickness of cartilage. And it's based on two main pieces of technology. One being a proprietary material, which has cartilage-like properties, and then we're actually 3D printing that material into a porous, organized mesh structure. And so the implant is designed to have cartilage-like properties, so it actually dramatically shortens a patient's rehab because you actually are replacing the damaged tissue with something that is load-bearing. But at the same time, the combination of this unique material with this 3D printed porous structure actually recruits stem cells from inside the patient's body and then directs new cartilage formation over time. And so we're looking to bring clinical value because we can dramatically shorten recovery, but then also we are growing healthy tissue, and this is how we're going to ensure a long-lasting recovery for these patients. And how do we know it works? We have done an extensive amount of preclinical testing to date. Most recently, we did a large animal study where we showed two main things. One, that we get an incredibly high quality cartilage that develops. And number two, we showed that this cartilage develops at an incredible speed, roughly half the time of even some of the most impressive technologies which have not yet been commercialized. And in terms of things that are actually on the market, really the standard of care is still surgical interventions and different types of grafting technologies. And so there's preserved cadaver tissue, there's live cadaver tissue, and then there's also approaches to actually culturing cells, basically providing a cell therapy to regrow the tissue. These types of treatments suffer from both poor short and long-term clinical outcomes. They also are not designed to functionally replace the tissue. They're just trying to regrow something. And so we're really looking at recovering patients within 4 months and then having something that, as I said, can actually provide a very high quality tissue that will last a long time. But in addition to that, there's a really important value proposition here. And that's all these treatments, whether it's tissue or cell-based, suffer from huge issues with efficiency and also cost. Many of them are actually still not reimbursed by payers because they have a anywhere from $10,000 to $50,000 out-of-pocket cost. And then in the case of cadaver tissue, it's very difficult to source that tissue. So you have patients waiting half a year to a year just to receive donor tissue. And so NanoCon would truly be a low-cost, off-the-shelf option which could be used as needed. And also a price point which would be within the range of what's comfortably been reimbursed by payers. I want to be clear that we have been working very closely with the sports medicine community since we founded the company, and even before that. And at every step of the way have taken both feedback and actively collaborated with sports medicine physicians. And they all say the same things, which are very encouraging for us, which is that they want to see something that does dramatically improve the short-term outcomes, as well as ensuring better long-term outcomes. But then they also really kind of echoed this problem with the existing treatments where they basically want something that looks like what a knee replacement looks like for the general orthopedic surgeon. They want something that is low cost, off the shelf, and can basically be a procedure that can be done at a high volume, so that they're both capturing some of this loss from inefficiencies in treating patients quickly, and then also have something which they know that the average patient who is going to be relying on insurance to pay will be able to receive the treatment. In dissecting the market, we have really looked at a very highly targeted patient population, and based on a projected price point of around $3,000, we estimate that this would be roughly a $1 billion market. But when you talk about starting to treat other joints, especially the hip, the ankle, the elbow, and the shoulder, you get close to a $4 billion addressable market. And then we also think there's a lot of use for this technology in other areas such as plastic surgery and spinal surgery. So we estimate a total $10 billion value for the technology as a platform. And looking more specifically at this patient population, our candidate patients will comprise roughly 371,000 people annually. Compared to what's done now, all of the cell-based therapy and all of the graft procedures that are done only amount to close to 5,000 patients. So there's also an incredible opportunity to expand the patient base. To talk about myself and my team, as I said, I'm the CEO of NanoCon. I have taken the company through a number of different accelerator and incubator programs, including JLabs and most recently the UCSF Roseman Institute. I'm also responsible for all of the fundraising to date, which has included grants and SBIR and some pre-seed investment. And I also have prior medical device experience with the medical device startup SonaStick. My co-founder and CTO is Dr. Nathan Castro. He is a very highly decorated researcher in the field and has actually also participated in a clinical trial for a 3D printed orthopedic device. We also have a really strong core management team, very highly experienced executives in the life science space. Matthew Shear is very notable and was also involved in the Sonostick project. Gary Wakeford as well is a longtime business development executive in the medical device space and has exited a company. John Rodriguez is our product development executive who also has prior domain experience with a cartilage therapeutic and has also exited several companies. And then Dr. Chris Canova, who is a knee reconstruction specialist, is our chief medical officer and is a KOL for Zimmer. And we also have in-house legal and IP counsel with deep domain expertise. We have a very strong clinical advisory board as well. These include some of the top names in sports medicine and cartilage reconstruction, physicians from the Stedman Clinic, Rush Orthopedics, and some top academic centers, and then also a physician for the United States military. So to talk about the current round, we are raising $3.5 million preferred shares. The primary use of these funds will be to conduct a first-in-human study to start to evaluate this product and continue on the PMA approval pathway. We also plan to use this funding for product development, manufacturing validation, and also to continue to grow our IP. And then we also are going to be able to pursue additional grant funding, especially once we have some clinical data. This will open up opportunities potentially at NIH, but definitely at the United States military. So to wrap up, I want to kind of talk about where the company has started and where we've gone from. We founded in 2016 with little more than a few interesting papers and PCT filed. Since then, we have raised close to $500,000, as I said, through grants, have significantly de-risked the technology by showing strong preclinical proof of concept. We've also grown the patent portfolio. We currently have 4 active families, and 2 of those patents have been awarded. And we're poised to move very quickly. At this point, we just need to finish closing this round to begin our first-in-human study. And then once we do that, we'll be able to move, again, very quickly along the clinical approval pathway, and we are projecting an M&A exit in 2024. So again, my name is Dr. Benjamin Holmes. We are NanoCon, and I want to thank you very much.
Ben, thank you very much for that presentation. I really enjoyed the energy and the passion that you brought to this, and I think it's a very interesting and exciting project. The question I have is, I know that mesh has been used in a variety of medical applications, much different than the application you're looking to apply it to. I was wondering what term that term has, what significance that term has to both medical practitioners and patients when they hear the term mesh. I know there has been some lawsuit and litigation over certain mesh products. How does that factor into your business?
Yeah, that's a great question. I think in orthopedics, it's generally not a liability. Porous and mesoporous materials and meshes and weaves are used for a variety of bone applications. So I think it doesn't necessarily have the same taint, as it were, as it does in the fields you're talking about. I would also say that maybe mesh probably isn't even it really isn't the right way to describe our product, because that implies that it's something that's woven together, whereas, we're using printing to actually create an integrated deposition of layers. So, I think probably, maybe that's a good point for the future that we should just
to move away from that term, perhaps. Yeah, and just kind of a follow up to that, what do you anticipate the wear and tear being? Like, how long do you think this the mesh, the woven fabric will last compared to what's been done before?
Yeah, I think that the jury's still out in a scientific sense, because we obviously haven't done a 2-year-long clinical trial yet. But I'll say that the fact that we are actually regrowing a high quality, healthy cartilage bodes very well for longevity. The things that fail on this 2 to 4 year horizon currently don't actually regrow cartilage. They regrow some kind of scar or something that isn't actually cartilage, but kind of performs some function. And then in terms of the material, we've done quite a bit of mechanical testing already. We certainly plan to do more as part of our product development, but we've shown that it's fairly robust and it's elastic. So there's a lot of strong evidence that it works together with the new cartilage that grows, as opposed to being like something that's static or something that's going to be like a contact surface. So I think that we've really de-risked that question. Although again, part of the path to market is going to be a long clinical trial.
Thank you very much.
Ben, this is Niall here. So on the longevity question, if we kind of flip it from your interviews, and I compliment you on your extensive interviews of the market, do you have a sense for what are KOLs, what are the market looking for in terms of, if you can say, we've demonstrated successful performance up to X number of years, like what longevity do they need to see to be energized?
Yeah, so 2 years seems to be sort of like the early adopter scale. You'll see things picking up once a product gets its 5-year indication. That certainly happened with a product called Macy that, they saw a spike in revenue once they were able to report an indication for 5 years. But that's after you've gotten the FDA approval already. That's just post-trial follow-up. So the product could be approved and on the market already. I do want to say, though, that through our engagement with potential strategic acquirers, they're very excited about the prospect of something that reduces recovery time. Nothing has done that. And that would be very significant. The KOLs really like the sound of that, too, because, the patient population that's getting their knee scoped is actually shopping around for surgeons, maybe at a higher level of scrutiny than in other fields. So, someone being able to say they do the NanoCon, which has you back to playing tennis and working and exercising in 3 months, is really compelling when everything else takes 8 to 12 months. So certainly, like, our strategic mentors really are excited about that. And I think that would also really be a big differentiator, in terms of market adoption.
That's a great answer. Thank you.
Thank you.
Yeah, thanks, Ben. Great presentation. For that return to work, is that something you're looking to fit into your pivotal study? And then, as an endpoint, and then beyond that, could you comment a bit more on the competitive landscape of maybe earlier stage opportunities like HiLX, who've raised a good amount of money, and how they fit into the greater cartilage universe?
Yeah, absolutely. So yes, you're absolutely correct that would be an arm in a pivotal trial that just would end early, and you'd have some kind of indication on recovery time. In terms of how this fits into other products that are on the market, I think HiLX is a great comparator, especially because that's a technology that actually tried to do these types of lesions and couldn't, and so they're actually pushing more for like an alternative to a joint replacement now, where it's like a better material than the high impact polyurethane and the metal that are in those types of replacements. So I think that we would actually fit in well in a large strategics portfolio with the HiLX technology.
Excellent. Thank you. And I think that's all the time we have for questions, and this concludes our program. So Brian Koblitz from GW will explain how to next join the Zoom networking sessions. So thanks, everybody.
Thanks.
That is all the time we have for today's pitching session. A very big thank you to our panelists, Charlie, Peter, Matthew, and Niall. I really appreciate your time and your insightful questions and comments. Another big thank you to all of our presenters today. You've worked so hard on these pitches, and even harder on developing these technologies to bring them to the patients that need them. Thank you again to our sponsors, Wilson Sonsini and Blank Rome, that helped make this event possible. We're now ready to move on to the networking portion of the agenda. We're very happy to meet all of the attendees live and in person in a Zoom room of your choice. you have questions for the companies, you can go to the company's room and ask them directly. The links are below this video. You can also choose a room of one of our sponsors and learn from their legal expertise, or we can just mix and mingle in the general networking room. On behalf of George Washington University's Technology Commercialization Office and on behalf of Georgetown University's Office of Technology Commercialization, I thank you for joining us for this inaugural DC Startup Showcase.